Now, I do like the fact that within the lecture, on the lecture, again, it's not much.
When it comes to discussion time, like
Other kinds of questions.
so
Strategy this year.
No, so it should be.
It's part of
Actually,
the lecture. Is the reservation.
I'll say because like I have you listed as in the discussion room. Oh yeah. So you can like if people type questions, you can respond back that way, or the why is this not? Or so there's also if people want to join a breakout room, and you can chat with them.
okay.
8 years ago.
Yeah.
So
Same
when
thing as like
when that time
an
comes,
equation.
it's
You
going
have
to be
to have to solve
Yes.
pretty
Yep.
complex math.
Excellent.
Hello, everyone. Welcome tonight to our third evening of Remaking Life, our synthetic biology soapbox series. It's nice to see some familiar faces in the crowd. Just so everyone knows, on your seat there was one of these flyers, and they list the upcoming talks. So this is week 3 of 4. So glad you could join us tonight. Hope you'll consider joining us for our last program next Wednesday night, October 21st at 6 p.m. So to go over tonight's format, it's going to be a little different from a lot of talks you've been to. The format for this evening is we're going to have 2 short talks by Kristalla Jones-Prather and Ron Weiss, and they'll be sharing their research with us. And each of those will be about 20 minutes. Then we'll have small group discussions at your table where you guys can talk to each other about questions you have. We'll also have Twitter fall running, so you can tweet in your questions. And then we'll come to a large group discussion where we'll be taking questions from our online community as well as from the audience and our tweets. So this is an opportunity for you to join a discussion and talk with some experts in the field of synthetic biology. So thanks for coming tonight. Our first speaker is Kristalla Jones-Prather, and we'll be hearing about some of her great research at MIT.
Mic set up.
So it's very nice to be here. It's fantastic to see a large turnout of folks who are actually excited to hear Ron and me speak. I came in and I told Ron I was going to go first, because if you've heard many faculty speak, you know that if you give us an hour to speak, we've got no problem filling that up. But if you constrain us to something significantly less than that, it can get a little bit tricky.
It's like night now.
Is it not on?
It's on. It's on. It's a little higher up.
How about now? Okay, and I'll try to talk like this. In typical fashion, I probably have more slides I can get through in 15 to 20 minutes. And my objective is not to make sure that you understand all the itty-bitty details, but to give a bit of an overview to put things into context, and then to have a few more scientific details for those of you who might be interested in some of the more minutiae aspects of the stuff that we do. So I wanted to start with actually some definitions of synthetic biology. This is a phrase now that's been around for a long time. If you look at some of the history of how it's been used, there's a lot of variety and diversity that goes into that. If you look back about 15 years now, there was a definition that popped up in Chemical and Engineering News, which is the official publication of the American Chemical Society, that defines synthetic biology as using the synthetic capability of organic and biological chemistry to design non-natural synthetic molecules. And I've underlined here non-natural and synthetic molecules that function in biological systems to emphasize the fact that initially some of the views around synthetic biology were about how do we actually make things that didn't come from nature look and act like things that came from nature. I think over the years, that definition and where you see many of the practitioners of synthetic biology has changed. Such as we actually talk about using biological parts and devices and systems. I have these buzzwords and quotation marks here, but it's really about getting those to do different things. So we're not so much concerned about things that aren't normally biological, but rather trying to redesign these natural biological systems for useful purposes. And what I'll talk about today, and I think what Ron will complement in his presentation as well, is how we're thinking about taking what biology and nature have already given us, and then figuring out with an engineering eye, how do we actually use that for purposes that are defined by us to be useful to society at large. I prefer a practical definition of synthetic biology that actually comes from a research center that both Ron and I are part of, which is called SYNBRC, or the Synthetic Biology Engineering Research Center. And we've always defined this as just synthetic biology being a discipline that encompasses a group of people that are trying to make biology easier to engineer. And for us in particular, we're both in engineering departments, and a key aspect of this is trying to apply engineering principles, things that I would have learned being trained as a chemical engineer, that Ron would have learned being trained in electrical engineering and computer science, but applying some of those principles now to biology as a substrate or as a material that we work with. And when we talk about engineering, this includes terms and practices such as design, and I'll talk about design, about modeling, I'll talk about that as well. I know Ron works a lot on design and modeling as well. But it really is about trying to characterize those systems that we build to be able to now think about them on a higher level, and that's called abstraction. So rather than worrying about all of the individual details in this pointer, what I'm concerned about is making sure that there's a connection between this pointer and this laptop that will make it work. Turns out that connection is not working for my laptop right now, but just go with the analogy and pretend that it is. Okay, and I'll point out that over the years, one of the great enabling technologies for us has been DNA synthesis. So now we have the ability to start with what is just information in a digital sense and transfer that information or transform it into physical material, which is DNA. So we no longer need to necessarily have those natural pieces of biology in order to get something functionally that comes from biology. Okay, so there are many different areas of synthetic biology, and I have just a couple of snapshots of papers from the literature that are representative of some of the ways that synthetic biology is used. One of these is in biological computing or programmability, so thinking about how do you actually control information flow and processing using biology. A classic example of this, and one of the hallmarks of synthetic biology that ushered in this era was a paper in 2000 that talked about something called a repressilator. Essentially, a biological system designed with modeling to give you oscillation. So the output would go up and go down and go up and go down and go up and go down. It was called a repressilator because it used these biological parts that we refer to as repressors. And there's been lots of work with oscillators from that. Here's now another example that came on a few years later, another one that came on a few years later, another one that came on a year after that. Okay, so these ideas about how do you have increasing complexity in your systems that still give you, again, an output that you're looking for, which in this case was oscillation. A different area is therapeutics, and I think Ron will talk a little bit about how you can use synthetic biology for therapeutic purposes. This is an example, again, a few years ago from Mark Fussnagger's lab, talking about being able to control how therapies are delivered by using a mouse model, so actually introducing genetic circuits, or these engineered pieces, into a mouse, and by simply applying a lotion on the skin, that provided the trigger that you needed to have this output that was being sought, ooh, my battery's gonna die, for biological purposes. There's a plug here. I did read a power pack in case that happened. So I will talk and plug in as I go. Now, one area in which synthetic biology has gotten a lot of attention is in thinking about this design on a much broader scale. And that would be towards extending this principle of design beyond simple circuits, or circuits that allow you to express a gene, or circuits that allow you to have oscillatory behavior, towards something that's actually going to affect the entire functioning of an organism, and in this case, the entire design of the genome. And my computer is going to go to sleep, and I have to wait for it to go to sleep so I can wake it back up, I think. I don't have all my slides memorized, unfortunately. One second here. I can't tell you what was coming up there. So that is a classic case that came from Craig Venter's group, from the JVCI, the J. Craig Venter Institute. And that you may recall from a few years ago, maybe, I forget what the year was on the paper, made a big splash. I actually remember being in Canada for a seminar, and I wake up in the morning and turn on the news, and the headline is, "Scientists Create Life." Okay? And there was a lot of splash about the fact that what the scientists had done was to start with a gene sequence that allowed an organism to have all the functions that an organism would have. And then again, relying on this fact that you have information that can then be transformed into physical material, they were able to redesign the entire sequence of this bacterial genome. And then with that redesign, synthesize it, put it back together from individual pieces that came from now this key enabling technology of DNA synthesis. And in doing so, get that piece of DNA to be able to function now in a natural system. Okay? So what you had was a chemically synthesized piece of DNA that could then operate or control the function of the cell in the same way that the naturally occurring piece of DNA could occur as well. And you will see a lot of literature, if you go back to the time that that was produced, that debates whether or not this was really creating life or not. But I think what everybody agrees about is the fact that this was really a tremendous technological achievement to be able to take something as complex as a whole genome and then be able to get that resynthesized and have it function again in a normal host. My entire computer is rebooting, but I got this pretty much memorized and seen, so I'll keep going. So I think the next area that will pop up, and so far it's just been these little snapshots, so you're not really missing anything, moves into the work that we actually do, and that's actually trying to use biology for chemistry. And that is using biology to be able to synthesize chemical molecules where we can, instead of relying on petroleum as a feedstock, we would be able to use something like renewable biomass and to convert that biomass into useful materials. So now you can start to see that when we talk about synthetic biology, there are a lot of different areas that it could be used, and a lot of people are coming from different disciplines to try to figure out, if I'm going to take existing biological parts and redesign them for useful purposes, then what is that purpose that I'm excited about? What is the thing that I want to keep going? So, well, that's still booting up. So we work within synthetic biology in an area that's known as metabolic engineering, where again, we're focused on using biology for chemistry. And the challenges that we have when we do this is the fact that we start with a cell. We are feeding in something like glucose, so a sugar that the cell likes to eat. And what that cell really wants to do with that glucose is to convert it into more of itself. It wants to take sugar and process that sugar. If you go upstairs, you'll see models of amino acids, which go into proteins, of DNA molecules. And the cell would rather take that input that we give it and make all these individual pieces that allow it to make more of itself. What we want to be able to do is to have that cell, instead of making more of itself, transform that same chemical molecule, that precursor, that feedstock, into something that's going to be useful for us. And the challenge that we have is that the cell is not motivated to do this. Okay? The cell has, as an evolutionary imperative, the reason the cell evolved to exist in the first place was to be able to make more of itself. And so we're fighting this constant battle between trying to keep the cell from doing what it naturally wants to do, and instead shuttling all of that mass, or as much of that mass as possible, into these new molecules that we're creating. And we think about doing that in two ways. One is that if there are specific ways that we can think about the cell using that sugar molecule, using that precursor, that we can get rid of, then we say, well, let's just take genes out, which now takes out the little machinery that would convert A to B, and B being something that we want. So we'll get rid of stuff that we think is not useful for us. But we also are facing the reality that some of the enzymes that we're using in order to get to these new molecules don't work fast enough. So they're not really moving at a speed that allows us to get to where we want to go with the kinds of metrics that we need to be able to make sure that we actually are doing something that's useful as opposed to just a simple proof of concept. Okay, so I think I've gone as far as I can go without my slides, but it looks like things are coming up now. So the challenge that we have is that we've got all this complexity. We've got this cell that wants to take the sugar and make more of itself. And again, our goal is to get it now to take that sugar and produce the molecule that we're interested in. I went longer than I thought I could go without slides. Okay, so let me keep describing, and I'll jump into the slides when the slides actually come up. Okay, so it turns out there's a way that nature already does this. Okay, so a lot of the pharmaceuticals, the therapeutics, especially the original ones that were developed, are so-called natural products. And that just means that they came from a natural environment. In many cases, there were plants. In other cases, there are soil microorganisms where these molecules are useful to be able to fend off invaders. So within soil, you have certain bacteria that want to keep other bacteria from invading into its space, and so it will make and then secrete these chemicals that will kill those bacteria. And if those bacteria it's trying to kill happen to be able to infect us, then we can take those molecules now and produce them on a larger scale and use them therapeutically. This is the story of penicillin, which was the first mass-produced antibiotic. And essentially, all of the initial antibiotics are in some ways either natural products or what we call semi-synthetic derivatives of natural products, which means that biology or nature gives you a certain amount, and you use chemistry to give you more of that. The first cholesterol-lowering drug, Mevacor, I worked at Merck, so this is my Merck bias. Mevacor is a Merck drug. That was actually obtained from a natural product. And for a lot of these natural products, they are what are called secondary metabolites. Okay, so primary meaning most important, secondary meaning second most important. And the way that the cells will make these molecules is that they will first start by, making a bunch of itself. So it takes that sugar, again, that we're interested in, and it converts that sugar into more of itself, but then it says, well, I've made enough of myself, I still have some carbon that's left, and as a result of that carbon that's left, I want to be able to convert now that carbon into a product of interest, okay? So we want to be able to do something like that. Instead of just saying the cells are going to take that sugar and just eat it up and make more of itself, what we'd like is to have now, again, a designable system. So we're still relying on this core principle of design and synthetic biology. And we actually want to say, well, let's do what nature does. Let's grow initially and then have some way of triggering a production phase. So we'll start off and say, well, here's now this competing activity where we'll allow it to be high. This is now growth. And then we're going to have some change to the system, and that's going to cause this competing activity to go down. And if I look now at growth versus production, that means that I'm going to let the cells grow for a while, then I'm going to allow the growth to stop, and then I want production to take over. Okay? Again, in more of the synthetic biology language, this is what we call an inverter, where we're looking for input to be low to have output to be high, and then when input is high, the output is low. And our goal here, as I was saying, is to mimic this natural precedent of secondary metabolism. Okay? So we've been working on a pathway to make this compound called butyric acid. It uses glucose in order to make butyric acid, but really what the cell wants to do is to take glucose, it converts it to something called glucose 6-phosphate, and normally that's what goes into, or I shouldn't say normally, ideally, that's what goes into the product that we want to make. But normally there are a couple of different routes that are primary ways that the cell will take that glucose and use it to make more of itself. So biomass is more of itself. Energy is a stand-in for molecules that allow the cell to actually have the energy that it needs in order to make more of itself. So we looked at the system and said, so how do we actually design now to allow first this glucose to be used in the way that we want it to be used, and then to have it trigger to selectively be used for making the product that we want. And I'll point out that although there is this other route that goes here, one of the things we have to do as engineers is to analyze the system and say, well, what components really matter? And for the piece that just disappeared, I'll bring it back up for a second. For this piece, what we knew from the literature, from work that other people had done, was that very little of this sugar goes here anyway, only about 10% at the maximum. And if it does go here, it can make some components of biomass, but not enough for the cell to stay alive. Okay, so from our perspective, that meant, well, let's just ignore that part, because the real interesting stuff that's going on is going on here. So now we have this design goal that says we want to have a system where we can trigger and first allow things to grow, and then we want to switch and allow things to make product. Okay? And we actually had 3 different enzymes we could think about controlling. And the question we had initially was, well, how do we pick? This is where the modeling part comes in. That's one of these core engineering principles, which is that we could actually build a mathematical description based on information from the literature to predict for us what would happen if we selectively removed any of these 3 enzymes. So what's shown here is now from left to right, decreasing levels of these enzymes. And what's on the y-axis here is now a prediction from the mathematical model of how much extra material we could have going into this new product. And this model told us that these 2 targets wouldn't do much good. I could knock them down by as much as 50%, and the response that I would see would be minimal. But if I focused on this guy here called PFKA, And that actually seemed to produce about a 50% boost in what I would have available to convert to my product of interest. Right, so we then end up creating this system where we say, well, okay, we're going down this way. That's the path that matters. I'm going to get rid of that just to keep things simpler. And then we created what we call a valve. Remember, I'm a chemical engineer, so we use terminology that comes from chemical engineering. And for me, this is about being able to open and close pipes, in my chemical engineering parlance, to either, if this is open, I'm allowing the material to flow this way. If it's closed, then I'm shutting off the flow in this direction. That's going to cause a buildup now here that can go to the molecule that I want. Okay, and without going into the details of exactly how we did this, I can show you the results, which says that we have a way of controlling this that can now, when we apply this perturbation or this inducer, my input, this will really decrease the amount of that enzyme that I wanted to knock down. It actually goes down well below the 50% that was in our models. And if I don't add anything to it, eventually it goes down, but not as quickly and not to the same level that it does before. And if I want to know that it's working the way I expect it to work, what I thought I was doing or what I intended to do was to pick something that would restrict the ability to grow. So I can then measure how fast the cells grow. And now as a function of down-regulating that piece, so closing that valve, I can see that as I close it more and more and more, my growth rate goes down. Okay, so things are sort of working the way I want them to work. The next thing is, well, what I predicted from my model is that the cells are going to stop growing, and I'm now going to have more of this substrate, we call it, this precursor, to go into the molecule that I want. So I can actually measure and see, does it give me what I want? And just, I won't go over the details of what these are, but essentially this is now our experimental condition where we're trying to change the amount of that particular metabolite, and we can see that it increases by about 3 or 4-fold when I've made all these different changes to the system. And the last bit is, does this actually give me more of the product that I'm interested in? And the first thing that we tested was just the ability to make that first molecule in the pathway. And this is now as a function of when we actually choose to close that valve compared to not closing it at all or not having the valve at all. And in the best case, we get about a 2-fold increase in both the amount, so that's the titer, this concentration that's here, and the yield. And the yield is how much of that material I started with was I able to divert into the pathway of interest versus having it go down towards the cell and eating it. The last thing I just wanted to show is the fact that we can keep going all the way to the product we're interested in, which is glucuric acid. And in that case, we got about a 40% boost in the titer and the yield, which actually happens to match pretty closely our predictions for how high we should be able to get things to go. So just to summarize this, what we're really interested in is using microbes, using biology, and re-engineering them to make a lot of different compounds. And although I couldn't quite talk through without slides the pathway that we came up to do this, the way that we actually make glucuric acid, and that's the target molecule we had, is not the way it's made in nature. So part of what we do is to figure out new ways to mix and match things from nature to get new kinds of functions. We have to think of new ways to do this. And so I talked about now having this device that allows us to have this bottom-up design to be able to regulate flux in a particular way. And for us, synthetic biology really gives us a framework and a way of thinking about how can we use engineering principles to affect biological systems. And for us, that really means working within the context of metabolic engineering when we're focused now on these metabolic pathways. So I'm going to very quickly say my last thoughts is that synthetic biology, hopefully you saw from all the different examples earlier before my laptop died, that this is a very diverse field that has people who come from different backgrounds that are working on a number of problems with a number of different approaches to them, but everybody is really trying to figure out how do we make biology easier to engineer. So the last slide I just want to have is an acknowledgement that recognizes in different colors here all the folks who've worked on different aspects of this work over the years, and to acknowledge the National Science Foundation who has funded this research through a couple of different grants. I think I was right about 20 minutes. All right, thank you for your patience. You know, I always say if the slides disappear for too long, the only thing I got left is interpretive dance. And so I think we're all happy that it didn't come down to that.
Thank you very much.
Can we do questions now?
So we're going to do questions after both talks. So welcome. We have some new faces that trickled in. Welcome to our third night of our Synthetic Biology Soapbox series, Remaking Life. Tonight we're hearing two wonderful talks. We just heard from Christala Jones-Prather about her research. And up next, we have Professor Ron Weiss sharing his research. I'm Jennifer Novotny, by the way, and I'm the programs coordinator here at the MIT Museum. I'm really glad you were able to join us tonight, and hopefully you'll be able to join us for our final part of our Soapbox series next Wednesday night. That's looking at more of the social implications of synthetic biology. It's called Who Needs Rules? And we have George Church and Kenneth Oye speaking. So it should be a very lively evening, and hope you guys will join us. And if you can't join us in person, please consider joining our live stream, where you'll have opportunities to ask questions to our scientists as well as have online discussions. But with that, let's hear from Ron.
Thank you. Yeah, thank you for inviting me. You can hear me fine. By the way, interpretive dance. So, did you go to our last? So we had, we belong to Synthetic Biology Engineering Research Center, and I think one of the highlights of the last retreat was interpretive dance of tumor-killing bacteria. So if you want to, I don't know if it's on YouTube. Maybe you should go search on YouTube tumor-killing bacteria interpretive dance, and if you're really bored, huh?
Neither of us dance.
None of us dance, so that's so it's safe to actually look at it. And so yes, I've known Chris for quite some time now. We have several grants that we share. We have a different focus in our research. So I focus a little bit more on therapeutic aspects of synthetic biology. So where we want to be engineer systems that we can introduce into mammalian cells somehow, and then would have maybe a therapeutic effect or allow us to understand the way drugs might interact and might maybe produce drugs in a better way, or understand ways by which drugs could actually affect you before testing them on people or testing them even on animals. So I'll actually give you a couple of examples of those things. I'm a computer scientist by training, and so for me, about 20 years ago, when I was a graduate student here at MIT in computer science, I all of a sudden became fascinated with the notion that we might be able to program cells in the same way that we program computers. And so I was happily making progress along in a normal computer science kind of PhD, and I realized one day that this is what I want to do. So I went and talked to my advisors, and they told me, "Oh, you're so close to getting your PhD that don't do it. You can leave it for later." And so as a typical MIT student, I did exactly the opposite of what they told me. So that's my one advice: is do the opposite of what your advisors tell you. I don't know how that works, because then you have to ignore me. But the point is that I then spent a lot of time in a wet lab, trying to understand what it means to actually program cells. And so for me, kind of the fantasy is this notion that we can treat a cell as really just something that we can program. So for example, something where we can write a high-level program, a biological program. So this is not going to be in Java or Python or Perl or anything like that. This is going to be a particular programming language that's suitable for programming cells. We'll write this program, it will specify some kind of hopefully interesting or useful behavior, and then we would have a biocompiler, for example, that would figure out how to convert that high-level program into some kind of a gene network. And then from that gene network, there would be a mechanism that would actually create a DNA molecule. And so you end up getting, for example, a tube that contains DNA. You put that tube into the cells, and then they do exactly what you want. This is kind of our nice fantasy. For those of you that have actually been in a wet lab, you realize this never actually happens. It never really works. And so it turns out that programming cells is a little bit more difficult than we had initially, or at least I had initially anticipated. But we want to, as a result of that, come up with what does it take to get us there. So what is the new engineering discipline that will actually make this happen? And so as a young scientist, this, by the way, is courtesy of Tim Lu, one of my colleagues, this is the way I used to think about a cell, right? This nice modular entity that we can do plug and play and put modules in there and connect them up and wire them up, and great, they do exactly what we want. I was sharing a cab ride actually with the first author, the first paper that Chris showed you about the repressilator. So this oscillator that works in bacteria. So we are sharing a cab ride outside of London, and so he shared his perspective on cells. It's a bit, more like this. I guess there's Mako Tacos nearby, so I don't know. So it might be, so if anybody's hungry after this talk because of subliminal messages. And so, what is a cell? Can we treat it like this? Can we treat it like that? So, it's not, I'm not really quite convinced. It's more like, the messy taco that you're seeing here. But it's not also quite as, modular and quite as clean as we'd like it to think. And then in order to make this thing possible, we really need to think about ways by which we can combine, for example, things like systems biology with synthetic biology that allow us to now achieve new practices that really respect this engineered substrate, really try to understand what is unique about biology that makes it different from any other existing engineering discipline. And so when we start to think about synthetic biology as a whole, this is a metaphor that we often use for the systems that we build. Okay, so this notion that within the system, the genetic circuit, for example, that we build, it's going to have first sensors. Okay, so this would be genetically encoded, and I'll actually show you a few examples. These would be genetically encoded sensors that we can embed into a cell using DNA. And they would allow the genetic circuit to understand what's going on either inside the cell, so for example, presence or absence of various biomarkers inside the cell, or understand what's going on outside the cell. And then the other portion of the genetic circuit would take the sensory information and integrate that in some kind of a genetically encoded biocomputer. Okay, so some kind of a control mechanism that works with biochemistry inside the cell. So we'll be able to integrate sensory information, make decisions about what's going on based on the existing sensory information. It may be also based on the past, maybe also based on the history. And after it makes these decisions, it would actually actuate. It would, for example, affect outputs inside the cell or perhaps even outside the cell. Okay, so this notion of sensing, processing, actuation is actually a typical way by which we think about building biological programs. And I think still it's a very useful notion, okay? And it's a very useful way to think about what we're actually trying to do. Over the last few years, there's been increasing emphasis on essentially respecting biology a little bit more. Okay, so trying to understand what makes a substrate different. So for example, rules of composition. So is it really the case that if we have one part, we have another part? Is it really the case that 1 + 1 is equal to 2 in biology? Can we really predict the behavior of a composite system based on the characterization of the parts? Which, it's not so obvious. I think we're making good progress on that, but it's not so obvious. Gene expression noise, cellular context. So for example, I put here, I'm not indicating to you here what kind of cell this is. So when I think about a genetic circuit and how it's going to operate, how is it going to be affected by putting into one type of bacteria versus a slightly different type of bacteria versus yeast or mammalian cells? Okay, so we don't have really a great concept of that yet. And we're working on it, but it's not quite there. Cell death or incomplete models. So this notion that we don't want to wait until we fully understand the cell before we actually start to engineer it, because I'd certainly be dead by then, and so we want to get started now. We want to come up with metaphors and mechanisms that allow us to engineer reliably, despite the fact that we have only an incomplete understanding of the substrate that we're trying to engineer. I'm not necessarily saying that we fully understand, let's say, laptop and how it fully works, but there's still really good understanding of how it works in terms of making certain applications work within that context. And so in terms of what we typically do, our typical workflow, we'd like to start with software, design things, and then understand how we can take biological parts, assemble them into genetic circuits, put them inside the cells. Also spend a lot of time on trying to understand if we can predict what's going to happen. So wouldn't it be nice if we had some notion of what's going to happen after we build a circuit, put it inside the cell, how we can make things more modular within that context, and then spending more and more time and then applying that to specific areas where we think synthetic biology can perhaps cause a paradigm shift on how we address specific problems. And for example, I'll talk briefly about cancer classifiers, about organs on a chip. I won't talk about vaccination, but those are 3 application areas where we think the notion of program cells can actually make a fundamental difference on how we approach these particular problems. Okay, and so the way we start is by having parts. So we'd like to kind of have this Radio Shack, or I don't know if it's still in business, by the way, but some notion of a parts registry that actually exists. You go to partsregistry.org. You can browse many parts that are available, actually on the order of 15,000 parts that are now available for you to obtain, as long as you're not some kind of a rogue country. And this parts registry actually exists here in Kendall Square, so you can send them an email and actually ask for parts, and they'll probably send you these 96 384 weld plates that contain all these genetic parts, and then you could go into your lab or your kitchen or wherever you want to do your experiments and to begin to put them together to create circuits. And so these parts include things that regulate activity inside the cell, that allows to get cells to collaborate by having cell-cell communication, other parts that allows to see what's going on inside the cell. So we basically have our debugging mechanisms, right? So that cells can tell us whether they're acting correctly or about their conditions, and also allowing us to interface with the cell, sensors and actuators. And so I would say the biggest challenge in synthetic biology, and perhaps even another definition, so you've heard at least 2 or 3 definitions so far, so let's continue to confuse you with additional definitions to give you a true state of the field, is that how do you create functional systems out of perhaps well-characterized parts? Okay, so that's yet another definition of synthetic biology. And so in order to do that, again, we'd like to have this notion of compilers and software mechanisms. So this is work with Jake Neal and Doug Densmore, starting with high-level descriptions and then having them be driven down to something that's actually DNA that we can put inside the cell. So I'll just give you very briefly, so we can you can actually go to this website right now for any of you are interested in writing code, register for free, and then you can write this high-level code, and then you won't get DNA yet, unfortunately. We hope in the future maybe we'll actually be able to provide DNA for it. What you will get is a MATLAB simulation that allows you to see what might happen when you actually perhaps build that particular genetic circuit. Okay, and then so this is the kind of code that you write. Unfortunately, it's full of parentheses. I don't know if people here are familiar with writing code with parentheses. Anybody here? Yeah, Lisp old-timers. Cool. Glad to see. I think there's more of you in MIT than anywhere else in the world. So it's good to give talks to you guys. And so for those of you that don't know Lisp or anything like that, or don't like a lot of parentheses, this is a simple program. It says if some molecule is high, make a cyan fluorescent protein, else produce a yellow fluorescent protein. So it might be a program that you want to write. And so the biocompiler will go through a series of steps that I'm not going to talk about in detail, but it's automated. And so it goes data flow, and then an abstract gene regulatory network, and then an actual sequence of DNA that might implement that high-level program, and then robot assembly instructions. And then we actually have a robot in the lab that right now is a $150,000 robot that's basically collecting dust. We figured out that if we got that robot to really work, it would bankrupt the lab in about a week or so. So it's too expensive for us to run. But the idea would be that there would be a robot that actually would build this for you. In fact, at MIT, another Chris Boyd, has set up a really nice $30 million facility that will do all of this assembly. So build thousands of genetic circuits a day. It's crazy. What we hope to do is actually make this available to everyone. So we have this collaboration with Pete Carr at Lincoln Lab to do it with microfluidics. So it's something like on the order of $30,000 that we might be able to have on every graduate student's bench, and actually won't bankrupt the lab that's actually trying to build these genetic circuits. And so maybe there'll be full automation of this, so you won't have to worry about how to build genetic circuits. You said, just what genetic circuit do I want to build? It gets built for you. You put it into the cells, and then you realize it doesn't work, right? Okay. And so you can write high-level programs. This is another example of a high-level program, a little bit more complex, and it starts out with a rather, again, complex-looking genetic circuit, and the biocompiler can then take that and optimize it to the point where now it's smaller and something that would be more reasonable to actually build in the lab and then actually test out, or maybe make. Again, this is to the point where you can imagine making 100 different versions of this, or maybe even 1,000 different versions of these. Maybe you want to know ahead of time which one of those is actually going to work. You build all of those, and then you put them into the cells, and then you figure out which ones actually give you the right behavior, which is slightly a different way of thinking about building, for example, cars or computers. You don't necessarily build that many of them, test them out, and then see which one of those actually work for you. And so, what do we want to build? So if we can build circuits, what kinds of things might be interesting to actually build? And so we do things. We spend a lot of time building things that make fluorescent proteins, fluoresce in different colors, or go up and down, or go this, go that. Spend a lot of time doing that. But again, over the last now 10 years ago, we've been trying to focus also on specific applications. Okay, so besides just turning on fluorescent proteins, what kinds of other things might we be able to do? And one of those application areas is for cancer. So in particular, one of the biggest, if not the biggest challenge in cancer therapy is how do you create a therapeutic agent that is highly specific, that essentially has no false positives or no false negatives. So it can go into cells or go near cells and then figure out this is a cancer cell, this is not a cancer cell, and if the answer is yes, kill the cancer cell. If the answer is no, just basically don't touch it. Okay, so if you can have something that is that precise, that might be a significant step forward towards actually effectively fighting cancer. Okay, and so the idea here is that most existing therapies are trying to find out what is the biomarker for that particular type of cancer. What single molecule or cell surface receptor might distinguish that particular cancer cell from healthy cells, for example. And the problem is that oftentimes, if not perhaps almost all the time, that doesn't exist. Okay, there's not just a single biomarker that can completely distinguish between cancer and healthy cells. It's a complex problem. It's a complex system. And so we kind of are trying to, again, the notion of respect biology a little bit more. So we're saying, okay, we can't find a single biomarker that can distinguish, but we can look at multiple biomarkers with synthetic biology. And by looking at multiple biomarkers, all of a sudden we can actually make highly precise decisions. Okay? And so this notion of building a therapeutic agent that can do now a complex computation inside a cell, and based on that complex computation, figure out, is this a cancer cell or not? Okay? And so what is that complex computation? It's this program right here. Okay, so a high-level program, detect and kill. So HeLa is a particular type of cancer cell. Okay? Cervical cancer. And so the notion is that we write a bioprogram that looks at microRNAs. Okay, so microRNA is a specific type of RNA. It's a regulatory kind of RNA. You can use that as a biomarker. And specifically, we did a computational study and realized that if a cell has this particular profile of microRNA, okay, where this microRNA is high, this is low, this is low, and this is low, and this one is high, and this or this is also high. Okay, so it's a particular profile. It's actually a pretty simple profile, by the way. It just says these are low, this is high, and one of those is high, or both of them are high. The point is that if the cell exhibits that profile, it's a HeLa cancer cell and no other cell type. Okay, so that's based on the computational analysis of what's known about microRNA expression. And I would also argue that any cell type would have a unique microRNA expression profile. Okay, so we potentially can do that for any cancer cell type or even any cell type at all. Okay, for other purposes even besides cancer. Okay, so the point is, if this profile is matched, kill the cell. If it's not matched, just have the therapeutic agent go away. Okay, so how does that work? So just very briefly, I won't go into all of the details. So we first start with this is a DNA sequence, and we say, we create a situation where this is the output protein, and if this microRNA is present, or this microRNA is present, or this microRNA is present, they repress the production of the output protein. Okay, that's what this genetic circuit is saying. And so the notion is this output will be high only if this is low, and this is low, and this is low. It's like this logic operation, which is an AND gate, a 3-input AND gate, with 3 NOT gates as inputs. Okay, so we converted this biological problem into a logic circuit problem. that this satisfies a portion of it. Okay. Again, without going into all the details, now this branch right here allows to extend this AND gate. So now it's a 4-input AND gate, and this operation basically microRNA basically does the not operation. So the not operation allows us now to connect that as yet another input. So it's required that this microRNA is high in order to allow expression of the output protein. So now we have a 4-input AND gate, and we can then further add another 2-input OR gate, which is now connected to the AND gate like this. So now we have a 6-input logic function that only turns on in HeLa cancer cells in no other cell type. Okay, so if you want to talk more about this genetic circuit, obviously it's complex. This is something we can discuss later. And so we have data, this is usually in Petri dishes, showing that we can get expression, high expression in the HeLa cells. In the other cell types, it's low. Okay, we can actually also selectively, we have a Petri dish that has these multiple cell types, and it selectively kills the HeLa cells, and it basically doesn't do anything to the other cell types in the Petri dish. Okay, and then afterwards we started to work on it. So these are just preliminary results to begin to work on a mouse model that actually has a breast cancer in it. Okay, so these are some experimental results showing mice with breast cancer where the notion is that without treatment, the cancer actually grows, the tumor actually grows over time. Okay, and then this is not. These are data not from the full therapy. This is just the beginning of the therapy, where we just put an output protein that modulates the immune system, causes the immune system to come in and fight the tumor. Okay, and so we're showing even without that fully complex circuit, this is the normal tumor growth. This is actually melanoma here. So this is breast cancer. This is melanoma. Normal tumor growth, and then what happens when we have not even just the beginning of the therapy. So it's able to really reduce the tumor growth significantly. And so the hope is that once we put in even the entire circuit, we would even get better performance than what we're seeing here. But we already have exciting data with this. Okay. And the last thing I want to briefly talk about is this notion of organs on a chip. Okay, so this notion that we take skin cells from a person, okay, de-differentiate that into basically what are stem cells, so called induced pluripotency stem cells. So they essentially act as stem cells, and then we can take those stem cells and then program them to differentiate into these organoids in a Petri dish. Okay, and I'll show you data on that briefly later. Why would we want to do that? Why would we want to take your skin cells and then grow organs on a Petri dish? So one idea is for drug development. Okay, so the way drugs are developed right now is they have specific molecules, and then they test them out, for example, on mice and rats that are probably not that genetically related to you. Wouldn't it be nice if we were able to actually grow a mini organ that is genetically identical to you? So for example, a liver on a chip, that is basically your genetically identical liver on a chip, put drugs on it, and then figure out what's going to happen to your liver ahead of time. Okay, is this going to somehow have some toxic interactions with your liver? Wouldn't that be nice to find out prior to actually taking that in? Or what happens when you take drug combinations? So a lot of people now, especially as you get older, begin to take more and more combinations of drugs. So if you're taking 10 different, 15 different drugs, the pharmaceutical company is not going to test that combination for you. Wouldn't it be nice if you had maybe in your, I don't know if your kitchen freezer or fridge or anything like that, your liver on a chip, you say, okay, let me put those combinations of drugs there, and then have that tell you what's actually going to happen ahead of time. So maybe transform the way drug developments are done, and then maybe in the future even do organ transplantation. So actually remove the issue of an immune system reaction, because this is going to be actually your genetically identical liver. Okay, so we write computer programs. I'll just show you pictures. We write computer programs to genetically program these cells. Okay, and so we started to program a liver on a chip, And so this is the beginning of that program. We took these induced pluripotency stem cells. Initially, these are basically these iPS cells in Petri dish. There's not much going on. After 5 days, they begin to make all of these patterns. Okay, they begin to make the different germ layers: endoderm, mesoderm, and ectoderm, and they begin to self-assemble. Let me see here. After 14 days, they begin to get liver function. So for example, this is albumin production, this is liver function. They begin to create vasculature, okay? And then actually within this vasculature, we saw the emergence of hematopoietic progenitors. These are basically precursors to blood. So actually kind of as a side effect of this is that we have a liver bud, embryonic liver bud growing in a Petri dish that's making basically blood cells for you. We've been able to take them out and actually mature them. So that they actually seem like pretty reasonable candidates for a blood supply. So it's kind of a side product, byproduct of what happened. We didn't really expect it. And another byproduct was that we were getting these structures to form from the liver that's growing on a Petri dish, and they turn out to be brain buds. Okay? So we have now the formation of liver next to this forebrain. By the way, this is these are iPS cells from George Church, who's going to be coming here. So presumably these are pretty smart brain buds. You could tell them about that. We haven't tested them yet. Their IQ yet, but I don't know. This is what mini George brain looks like, by the way. These are kind of growing from the surface. So yeah, and again, they form. So we have the formation of an actual liver bud next to brain with all the cell types that are actually observed in a liver bud. So just to kind of conclude, what we do is we start with some platform technology. How do we improve our ability to make DNA? For example, figure out ways by which we can be better at prediction, better at design, and then figure out specific application areas where we think the notion of programming cells can somehow have a fundamental paradigm or a paradigm shift to the way you approach this particular problem. So a lot of people to thank funding agencies, and I do want to quickly advertise. So we have an online course that we're going to offer starting January 4th as part of the MITx platform. And this will be free unless you want to sign up for $49, and then you get a certificate. I'm not sure what it means, but it costs $49. But I'm not planning on getting rich from this, by the way. But this is one of my colleagues, Adam Markin, that I'm co-teaching with. And I'll end by thanking you.
Thank you very much. So I hope you enjoyed hearing from our two synthetic biology experts. So now this is the point of the night where you guys get involved. So we're going to have small discussions at the tables. So you guys will discuss with each other. We have some iPads going around, and then you'll be able to tweet your questions, and we'll have a Twitter follow going so you can see what other people are talking about. And we'll do this for about 20 minutes, and then we will have a large group question and answer with our speakers. So happy chatting.
I think yeah. Yeah. So to design structures. Yeah. So I think the two kinds of structures, one of them is this notion of tissue structures, like the one that you're seeing there. Can we create all kinds of tissues by design? So you get to choose the identity of the cells and the location and the spatial organization of the cells, and maybe what extracellular matrix they're making. So that's one type of thing. We also have another project that got funded recently. We are taking Christina Teague, she's architecture, but I can connect you with that project. It's between my lab and her lab, and there we're actually engineering genetic programming bacteria to secrete cellulose as a mechanism to actually create also biomaterials in the structure. Yeah, so if you're interested in that, I can connect you with the right people in my lab. Yeah, I Okay. Okay. Yeah, that sounds good. Something for co-advising? Yeah, that sounds like fun. I mean, certainly, I think mesh as well with the general kinds of things that we do in the labs. Yeah. So yeah, I'd be very happy to talk to you about that. We're actually even in the process of putting together another proposal on creating these three-dimensional tissues, where it really is the architectural design is the motivation as opposed to any kind of real biological function. So this is something that I'm working with. One of the people I mentioned is Jake Beal, is a collaborator of mine. We're trying to put together, if you reach out to me soon, I think you can into that process. Yeah. Oh, okay. Yeah. Okay. You look very familiar to me. Okay. All right. Oh, yeah. Yes, I think there's a lot of synergy between systems and synthetic biology, and I think they're also fundamentally very different. So I would say that one way people think about it, I'm actually not sure it's the right way. One way people think about it is kind of the notion of systems biology being top down, and synthetic biology top down kind of understanding of cells. Synthetic biology is bottom up construction of cells. But I actually think that the distinction between them is the understanding versus the desire to build something new. So systems biology, it really attempts at unraveling the biological mechanisms that control cells. So how does a network of interactions lead to biological phenotype? Whereas synthetic biology is synergistic, but it aims at actually creating the network and then allows you to control creating a new network and allows you to control the phenotype. Is that? Yes, yeah. Yeah, so I think that the synthetic biology right now, as you mentioned, is focused on a smaller scale biological network. But I think that's more as a function of technology as opposed to fundamental principles. So I mean, ultimately, we'd like to be able to create de novo large-scale systems. It's just not, it's out of the scope of what we can do technically. System biology is presented with a naturally occurring cell, and then tries to understand how to holistically decompose that in understanding what principles was, what interactions are relevant. But depending on who you talk to in cystic biology, sometimes it's like the whole picture, but sometimes they would say, well, let's actually create biological dynamics simulations that actually start maybe all the way from the basic interactions, and then say, well, this is, in some way, what phenotype arises from those interactions. What's that? I see. No, I know about robo. I don't know if that's related. Yeah, I don't know if that's related to. Yeah, okay. Yeah, but I didn't know he was actually interested in using my system. No, I don't know. No specific. One of my former roommate in the office mate as a graduate student does something pretty similar. So she is also doing robotics, but she's doing this kind of self-assembling robots. I don't know if you've ever seen super cool stuff by Radhika Nagpal at Harvard. She has these programmed robots that somehow all interact locally, can create all kinds of interesting techniques and behaviors and structures and so on. So it's very cool stuff. Somehow, because I think the robot soccer, you saw it's interesting. This her work that she gets to being able to embed simple local rules of interaction between these computational robotic elements that then give rise to interesting global behaviors. Yeah, she may be on. Yeah. He's teaching the last lecture. It's completely open. You can check out that online version. You can come to the
All right, guys. Sounded like we had some really nice discussions. Seemed like a very lively group out there. So now we're moving into the large group discussion, question and answer session. But before we get to that, a lot of you guys just received a survey form. So the surveys help us make our soapboxes better, as well as find out if people actually enjoy them. So please take a moment or two to fill out the survey. If you notice, the last question asks for your name. And if you leave your name, we do a raffle at the end for this plasma bulb nightlight. So just as we are learning about how we can make organisms do some interesting and unique things, this light bulb, instead of being a typical light bulb, is actually a plasma light. So if you turn in your survey, after all of this, we'll do a quick raffle and somebody can be going home with a fun plasma light bulb. So let's get started with some questions. If you guys want to come on up. Unfortunately, our speakers will have to cooperate and share a microphone. So the way that it'll work is we'll take questions from the Twitter feed, and we'll also take questions from the audience. So you can raise your hand, we'll bring around the microphone, and you can ask that way as well. But why don't we start with a question from our Twitter feed? So let's start at the top. Could the cells learn and program themselves to recognize each type of cancer, i.e. cell learning?
Is that what we
Yeah.
I think it's possible. I think that that might be a future enhancement. Our notion right now is to try to really be kind of very precise and more predictive and take, for example, biopsies and be able to analyze them very carefully and then construct these genetic circuits that would correspond to the specific, let's say, biopsy or the specific cancer type that we found. I think the notion of learning is a very interesting notion. You have to be able to understand how to create kind of a genetic learning algorithm where, for example, let's say a genetic circuit kind of evolves to create newer versions of itself, and then the newer versions of itself somehow have a fitness advantage and allow it to then survive and then proliferate and maintain function. So I think if you can figure out what those specific, for example, fitness advantages could be, the learning part is actually doable. So the notion that you can take DNA and encode even within the DNA proteins that then manipulate the DNA kind of in real time and reshuffle genetic components, we can do that already. So it's really more about figuring out what the learning algorithm is, as opposed to understanding the biological mechanism.
Thank you. Again, if you have questions in the audience, please just raise your hand and we'll bring the microphone.
So my question is it feasible to take genes for something like a protein pump? Like let's say someone has cystic fibrosis and you want to give them a better quality protein pump on the surface of the cell. Can you take that gene and just put it into a virus and put it into the person and have it get expressed in them, or is that like naive?
Yes.
So why don't they just do that?
I'll answer.
That go from the pie in the sky.
So that's gene therapy, and there are lots of people who are working on this, and there have been a number of efforts to get this to work. And some challenges in getting it to work as well. So there's a very well-known case of, what's Jesse's last name?
Yeah, from
Of
UPenn.
a young man who was in a clinical trial at UPenn, who actually died after having a massive reaction to being infected with one of these viruses that was actually carrying a gene for gene therapy. So there are a lot of challenges that are associated with it. One is how to actually have materials that are able to effectively introduce that gene, and to do it in a way where you have something that can evade your immune system so that it's actually effective without actually triggering this very large response and ends up being more damaging than not.
So couldn't you just like pump the person full of like antibiotics and antifungals, and then just repress their immune system all the way down for like an hour to do it, and then get out?
Yeah, no. Ron's the yes, I'm the no.
Well, okay, so when I said yes, I was expecting Chris to come in and bring it down to earth, and a little bit,
All the way down.
all the way, little. It's obviously very complex. I mean, when I said yes, that I was going to get into some of the notions of why it's difficult, what the immune reactions that you might have. I think that it's an exciting future. So it's not the case that we can do it all right now. So for example, with DNA, one of the questions is if you have a virus that's a DNA-based virus and it goes into cells, then one possibility is that DNA will actually get integrated into the chromosome. And as a result of that, it integrates into the wrong place. It could actually cause cancer itself. Right? So one of the things I think that is actually exciting about synthetic biology is that we would put maybe the expression, the production of a particular gene within a delivery agent, such as a virus or nanoparticles or something like that. But we would also provide other features that perhaps increase the safety. So a lot of things that we're working on, that other labs are working on these notion of safety and kill switches. So the notion that you deliver a genetic circuit into a human, and if something goes wrong, we have ways of turning it off, or ways of actually having the DNA, for example, self-destruct. So just say, oh, the physician would see, the circuit is causing, the DNA is causing some immune reaction, or is somehow bad for your health. Then you take some kind of small molecule, and that small molecule destroys the DNA. So we actually can do that right now in the lab. We haven't tried that on human beings, but maybe, I don't know if that was in your sandwich. No, it wasn't. Okay, but
You're not experimenting on me.
no. Yeah. Formally, yeah, I'm not. So the point is that we are working on mechanisms to go beyond simple expression to improving the safety aspects, the control aspects, maybe the cell type specificity, so the protein only gets expressed in the right cell type, making sure that the protein only gets expressed at the right levels, monitoring the immune response to it, being able to modulate the immune response. So actually the thing that I showed you with cancer here, some time ago, actually was not a direct kill of the cells, but rather it was an immunomodulation. So it was a mechanism where we sent a message to immune cells to come in and actually fight the tumors themselves. So I think the notion of modulating the immune system is just tremendous. We didn't invent that. This notion of, for example, cancer immunotherapy is perhaps one of the most important kind of developments or inventions that are taking place right now in the field of biology or medicine and so on.
So I think kind of with that, one of the questions from Twitter is, how are the genetic parts inserted into the tumor cells to trigger this type of destruction or this immune response? I think it's kind of a more technical question.
Do we switch on? Oh, okay. Okay, so the genetic part, there are multiple ways by which we get genetic material into cells. So we mentioned the notion of genetically engineered viruses. That's one of the ways by which we do it. We also use nanoparticles. So these would be, for example, lipid nanoparticles. I have a collaboration with Daryl Irvine at MIT. So we create genetic circuits. We create the DNA. We give it to him. He creates nanoparticles, and then these nanoparticles enter the cell. Interestingly enough, we can actually even make RNA that is just RNA without any kind of encoding, any kind of vesicles or anything like that, inject that into muscle, and some of the RNA is actually taken up by cells. So we have bioluminescent mice that all we did is inject RNA into them. And the cool thing about that particular RNA is that it's self-amplifying. So all you need is one RNA molecule goes into the cell and is able to then make copies of itself, and then persist in mice for, we've had it even 3 months or so from a piece of RNA. The other thing that's really nice about RNA is that in theory, it's a lot safer because the RNA cannot integrate into your chromosome. So it never becomes DNA. So, in terms of, a safer mechanism for human, gene therapy, RNA is actually one of the exciting areas right now.
Question from the audience.
So first of all, thank you very much for the inspiring talks. I just have a follow-up question on the question asked here. So in animal experiments or human applications, these nanoparticles or circuits will enter all the cells?
No, so that's a great question. And one really important consideration when you think about these therapies is what is the delivery efficiency. And depending on what you do, whether these certain engineered viruses or nanoparticles or even just naked RNA, you get very different delivery efficiencies. They can be 0.1% of the cells. AAV, adeno-associated virus, is another mechanism that's considered actually right now to be extremely safe. So it's a DNA-based virus that's considered to be extremely safe. And depending on the tissue type, you can get penetrations or infections of up to 50, 80% or so. Now, that's when I mentioned this notion of immunomodulation. So in particular for cancer, we do not expect to reach every cancer cell. And so what we do is we go in there, we figure out, the genetic circuit figures out, is this a cancer cell or not? And if the answer is yes, what it tries to do is actually recruit the immune system to come in and take care of all the tumor cells that were not infected. So it basically says, oh, here's a tumor cell, signals for the immune cells to come in, they come into the right place, and then actually what it does is it kills the cell in such a way that causes the immune system to then recognize what kind of cells it needs to actually kill. So a lot of it is about interaction with with other cell types in the body to get the right action, the desired action.
So another question from Twitter. Can you tell us about tolerance levels when composing bricks? Is this like voltage levels when building systems from transistors?
So tolerance can be used in a number of different ways. So I'll talk about it a little bit in the context of chemical production, where when we talk about tolerance, we're really asking the question of how stressed is the cell as you add these different components to it. And it turns out that that's a very difficult thing to predict up front. Instead, what we have to do is do the experiment in terms of adding in these different pieces of DNA that encode for these enzymes, and then seeing what the impact is on them. And there are some cases where the tolerance is very poor, meaning that if we actually try to express a foreign gene, the cells become very sick. They don't grow very well. If they don't grow very well, they don't produce very well. So I talked about it in my overview, trying to switch between growth and production. It turns out if you grow too slowly, you also have a problem with production. So you have to balance this. And the perfect case is one where you initially have fast growth, because that's what actually gives you this capacity to do the synthesis that you want to do. And then you switch to something that's slower. So we can have cases where just the protein itself can cause a problem with tolerance. We can also have cases where what we're expressing causes other things the cell needs for survival to decrease, and that causes an issue as well. And then for a lot of the products that we're interested in actually using biology to make, one issue is that the product we're interested in making itself causes a problem as far as tolerance is concerned. So we actually have a difficulty of having the cell make that molecule without it killing it. So one example of that would be a lot of the biofuels that are produced. You never hear people talking about having to have disinfectants to keep gasoline from going bad, because it turns out that it just naturally kills most biological life. right? The same thing is true with ethanol. We use ethanol as a disinfectant. So there are certain strains that make lots of ethanol. There are others where you try to make a lot of ethanol, it actually kills the cells. There are a number of different things that we have to deal with in terms of being able to tolerate what's happening with the cells. And I think a useful thing to keep in mind is that we talk about designing these different circuits and pieces and things like this, but they always go into some cell that has its own background, metabolism and activity and things that it's doing. And as much as we try to have what we design interface with that to a minimal extent, such that we could have what we would call an insulated design, we're still relying upon the base capacities of the cell. And it is important to try to understand as much as we can what negative impacts the changes that we will have on that baseline activity, because that can end up having a negative impact on what our ultimate objectives are.
Question from the audience?
Yes, that's a question for you, Chris. Why are soil bacteria so good at making what they make?
Yeah.
And why can't we grow them in the lab or get them to make what they are so happy to make in the soil? Why can't we do it in the lab?
Yeah, so a very short version of an explanation for this is that in the environment, life is going to evolve to deal with the circumstances at hand. And the reason you find these soil bacteria that are so good at making these molecules is because they need them in order to survive. There's competition that's happening in this environment. And the competition says, if I allow my neighbor to grow completely unchecked, it will subsume all of the resources that I need for growth. So in order for me to survive, I have to be able to protect myself against them. Okay? Now, part of the catch there is that in saying that they do this well, they do it well for that environment. And the answer to your question then becomes twofold. One of the challenges we have with getting a lot of these organisms to grow in the lab is the fact that we're now introducing an environment that's very, very different from the way life exists. And I'll give you actually an example from something very recently that came out of my own lab, just a group meeting on Tuesday, where there's an organism we're working with that was isolated from the environment. And I described to you these E. coli cells that love eating sugar. It turns out that this organism, if we give it too much to eat, will essentially eat itself to death. Right? So if we give it a lot of sugar, it's not that it doesn't grow, it grows really fast and then it dies really, really fast. Because there's something that we don't quite understand about why it can't quite tolerate that much. It gets so excited. It's like you put a kid in a candy store, tell them to go crazy, they're gonna go crazy, right? And so there's a disconnect or a big difference between the natural environment where those cells have evolved to be at their optimal and what we want them to do. We need them to grow in a defined system. We often don't have the right nutrients to match exactly what's found in the environment. And that organism in the soil makes enough to be able to be optimal for its survival, but that production level is not nearly enough for us to be able to make enough of a compound to use it to actually produce a pharmaceutical out of it.
Let's take another Twitter question. So are iPS, I'm assuming induced pluripotent stem cells, modified with the genetic part in order to differentiate into a liver?
Okay, so in short, yes. Again, one of those simple responses. But so the notion is that we not just even create a genetic part, but we actually create a genetic circuit. So this would be so usually, one distinction is that a part, you can think about it as performing some basic function, and then you take parts and you put them together to create a circuit, and so the circuit now has a more sophisticated function. And so with respect to what we do with iPS cells, a part might be, let's say, make this particular protein or make this particular gene at this particular time. That might be a single part. One of the things we would like to do is create more sophisticated programs. So not just make this gene, at one point. So this gene would be the magic bullet that takes iPS cells and creates liver out of that. But rather think about kind of an embryonic program or a program of differentiation, where if you think about embryogenesis, what happens is not just a single gene that will take a stem cell and cause it to become a liver cell, and another gene that would take the same stem cell and cause it to become a heart cell, right? It's really something in embryogenesis. What happens is they go through a complex sequence of steps, right? As they differentiate, as they grow next to each other, as they create these more complex structures. So we'd like to be able to create something similar to that, where we actually create these genetic circuits, these genetic programs, integrate them into the chromosome of these iPS cells, and then say, okay, go ahead and make liver, but then so the way you make a liver is at a high level, you go from iPS cells to endoderm, mesoderm, and then to liver. But if you look at a developing embryo, right next to it, right next to the liver, you have the formation of a pancreas. And to the other side, you have the formation of a lung. So what we'd like to do is create a genetic program where we say, okay, go ahead and make endoderm, mesoderm, and then when the genetic circuit can detect that it reached that stage, using very similar technology to what we use for the cancer, so it can detect, and now this particular cell type is endoderm, then what it would do is it actually would bifurcate, and it would say, okay, the cells right over here become liver, and then send a message to their neighbors to the right to become pancreas, and send a message to the neighbors to the left to become lung. So that's kind of the genetic programs that we have in mind.
Good question from the audience.
So when I'm looking for a metabolomic pathway, the only thing I've used is EcoCyte, but it is organism specific. Do you know any non-organism specific? Because I'm really lazy. I just want to go, start with pyruvate, give me like a pathway to this. Is anybody working on that? Is your group working?
Yes, we're kind of lazy too. But we're inspired, and sometimes we're motivated to not be so lazy. So the short answer, if you're looking for established pathways, KEGG is the source that we go to. So KEGG is K-E-G-G, stands for the Kyoto Encyclopedia of Genes and Genomes. So it's actually a website that's based in Japan. And it's a curated collection of all the known metabolic pathways that allows you to search for some particular molecule you're interested in, and it'll give you a pathway for it that's considered the really a collection of all the known routes for that. And then if you're interested in a specific organism, you can actually say, well, show me what's known about how this pathway actually works in Acinetobacter as opposed to E. coli. And you can select that organism and it will tell you that. There are a number of different algorithms that are being developed if you're trying to approach metabolism slightly differently and say, well, what if I want to make some compound that's not found in nature? Or it's found in nature, but the way that nature makes it, as I mentioned before, what we did, the way that nature makes it is sort of complicated. Is there a different way to navigate all those different options for metabolism to get to something that's quicker? And there are a lot of these websites. EcoCyte has done this. The broader version of EcoCyte is called MetaCyte, so that's supposed to account for metabolism more broadly than just E. coli. They now have a tool that's supposed to help you with predicting pathways. KEGG is also supposed to have a tool for predicting pathways as well. I'd say most of them, for the most part, are still in the developmental stage, and none of them are really at the point where you could come in and just use them just as they are, and then get a single output that would be a reliable output that you would then go into the lab and try to execute. But we like tools and databases too.
So we are going a little over time, so let's go for 2 more questions, one from Twitter, one from the audience. I'll go for the Twitter question first, and then somebody can come up with another question. So what kind of math models do you use to predict gene expression?
So I'll give the simplified version, and Ron can give you the more complex version, because we're not trying to build livers, so we got fewer things that are going on. But we do look at trying to predict gene expression, and for the most part, we are interested in relative levels of expression. So we will build mathematical models that are based on our understanding of how gene expression works. We know that for us, ultimately, gene expression means some amount of enzyme with a certain amount of activity. So we need data from the lab that tells us what the parameters are for that activity. We also need data that tells us for the individual parts. We start with what's called a promoter, which is the point where the cell says, yep, I'm going to start here and convert DNA into RNA. And then we have ribosome binding sites, which are where the proteins, the ribosomes will say, yep, I'm going to start here and start making protein. And there's data that's available from experimental studies to show us what numbers match up with the rates at which this can be done. So we can take all of that and build ordinary differential equations and even include some noise if we want to reflect the stochastic nature of biology, to say, if I have this system designed in this way and this input, what's my predicted output from that? Is that sufficient?
That's perfect. I'm giving a lecture on this tomorrow, so if you want to come to my course.
What's the room number?
It's building 9057.
There you go.
There you go. Anybody's welcome to come in.
It was whoever wrote it on the Twitter feed.
Okay, then come on.
Do we have any more questions from the audience? Excellent.
Mr. Whitleys, or that was your last name.
Ron is good.
Ron said that you said that your approach is different and cheaper than the Chris's Voigt approach.
Oh, I didn't say better. I think it just
Can you elaborate on the difference?
that he has more money.
Are you saying, you're saying that robot will be out of the box?
Yeah, I don't know how much I can talk about his approach, but if you look right now at various, even companies out there, the notion of automation of DNA synthesis and assembly is just taking off tremendously. And so they're unnamed companies that I won't mention who they are, and people here in the room that have been employed in those companies, but they can make on the order of 20,000 or more plasmids a year using these liquid handlers that basically, these are robots that basically pipette liquid from here to there. And so that's what they do. They pipette liquid from here to there, and that allows them, and they just don't stop. And that allows them to just create tremendous number of plasmids. The concern, with respect to doing it in my lab was that the volumes there tend to be to the point where the cost of the enzymes becomes tremendous. And so some of these enzymes, like ligase, per weight is much more expensive than gold or anything else like that. And so if you just do the math in terms of the volume, it just becomes ridiculously expensive. And that's why we teamed up with Pete Carr at Lincoln Labs, who's, by the way, I'll just do 2 for 1. And so Lincoln Labs, so Pete Carr is from Joe Jacobs, is a former member of Joe Jacobson's lab. And so to answer that for free, the advantage is that I'm working with Pete right now on this. But Joe Jacobson has also done, and continues to do tremendous things that related to DNA synthesis and assembly. And so the point there is that volumes there are, at least 10 times less. And that allows you to basically, hopefully do DNA synthesis on every person's bench.
Well, thank you very much, and let's give our speakers a big round of applause. Wonderful. If you guys have surveys to turn in, we can wait a couple of moments until we do our drawing for our awesome plasma nightlight. So you guys could do a survey quickly too if you want to try to get it. Thank you all for coming tonight. We really appreciate having you. We hope you'll be able to join us next week for our final part of the series, looking at society and who needs rules and who benefits from synthetic biology. Are there any last surveys out there before we do the drawing? All right.
That's one of those things.
Do you want to? Last call for surveys. 5, 4, 3, 2. I'll let you grab that. So the winner of tonight's wonderful prize is Diane. Diane, are you still here? Diane with a B? Yes.
Oh, I'm so excited. Thank you. For Harris.
We have one more prize next week, so come again and try your odds. Thanks for coming, everyone.